Okay, Rhizaria. These organisms get their name from the fact that they have pseudopods. They're basically a form of amoeba, but they have pseudopods that are really, really thin and long and extend out from the body. Rhizaria, hair. And so these include the cercozoans. These include foraminiferans and radiolarians. Those are the primary ones that you may have seen before. The foraminiferans, generally called forams or something like that, are incredible organisms. These are marine organisms. This picture is Ernst Haeckel, who we talked about before. Great scientist. Took a little bit of freedom in terms of his drawings, but he was really good at drawing. And a lot of these old plates he drew of thousands upon thousands of foramen shells. Each one of these is a different species. This is a tiny, tiny fraction of all the different species of foramens that there are. The shells have chambers, and as the creature grows, it adds more chambers. It uses pseudopods for motility. They're microscopic, but they're just kind of on the edge. You can, if you look closely, you can see them. If you filter some water or look at some sediment. Many of them have kleptoporoplasts, meaning that they're poroplasts that they nab a cyanobacterium or eukaryotic algae and kept the poroplast and keep them for an extended period of time in cytoplasm. These shells are, I'm trying to remember, it's calcium carbonate for these guys, I think. Fossilized readily. And a pretty fair fraction of limestone is made up of the shells of these things. And so they sink to the bottom. That produces the main bulk of the oceanic sediment. And that gets compressed and ultimately shows up as limestone. What that means is you can take a limestone from a geologist can take a limestone, know exactly where it fits into the stratigraphy, can treat it mildly with acid, separate out the limestone, and look at all the shells. And you would be amazed at the kind of information you can determine about the ocean environment that limestone is formed in from these things. The evolutionary history of these things is known in exquisite detail. Every species is distinguishable, and you know exactly when they occurred, so you can date the rocks this way. And the kind of oceanic conditions are known. And so how deep it was, whether they're planktonic or pelagic, what the water temperature was, what the salinity was. Why on earth would you care? If you're an oil company, you care a very great deal. Because these overlying rocks have a lot to tell about what kind of salt or oil deposits might exist beneath them. And so the oil companies put a huge amount of effort into studying these shells and knowing what's going on for prediction of where you can drill. Here's one alive. Oops. Those of you who have the textbook recognize this picture. This is the shell itself, and then these are the pseudopods extending out from it. This thing first appeared 100 million years ago, and so just as an example, if you have a strata and you see none of these things and then all of a sudden they appear and the conditions are appropriate, that line marks 100 years ago, and it's the early Cretaceous, and it's right at the Cretaceous, early to late Cretaceous boundary. Cretaceous, that being when the last of the dinosaurs emerged. Also in this group are the radiolarians. They look a little bit like the foraminiferans, but notice that there are all these spikes and things that are part of the shell and not the pseudopod. They have silica tests, so this is glass, not carbonates. I talk about these as like three-dimensional snowflakes. That really is what the shells look like. These are also highly abundant in oceanic sediments, and they're used in the same way. Here's an example, Hexacontium giganta. This is big, not that big, but it's big enough to see. The shell is in 3 concentric circles, and the cytoplasm, the cell is indexed. The shell is inside the animal itself. So the outer shell marks an outer boundary. But all of this stuff is inside the cytoplasm of the creature itself. There are a variety of spicules with these long spikes. Obviously, well, not obviously maybe, but those are probably involved in reducing grazing by plankton. And the other fine structures that stick out are probably related to buoyancy, keeping the thing from floating up and down really easily. These organisms typically have a cortex, the outer part of the inside of the cell is high in lipid, and they use that to maintain neutral buoyancy in the environment. These things make up lots and lots of the plankton that you get. They're primary producers, some of them. This is a really weird one, the serpozoans. These things are amoebas in a way and flagellates in a way. They have a flagellate, pseudopods that stick out the front. The shell is right here and it has an opening, and those openings have teeth. They're not teeth that they bite with, but they're to keep things out. And the pseudopods extend out from that into the environment to pull out things to eat. The shell is covered in scales. That's how you know what they are. And they've got these fibers that extend out like so. These things are also silica. These things exist in soil. Most of the organisms in this group are marine or freshwater. This is a soil organism. Wet soils, actually.